So good morning. My name is Jin Chan, and my mentor is Dr. Min Hui Lo. I worked at the Molecular Pharmacology and Chemistry Department of Memorial Sloan Kettering Cancer Center. Oddly enough, my department works with chemistry, but I was given some biology. My project is the expression and purification of PRDM2 and PRDM9. So, first of all, some important terms for you to know. To keep it in the back of your mind while I'm working on while I am presenting. Well, a protein is made up of amino acids, and the function is to make enzymes, antibodies, and hormones. Amino acids are the building block of proteins. A plasmid is a DNA molecule that replicates independently of the genome. Basically, it can replicate without chromosomal DNA, which is the hereditary information that gets passed on. Antibiotics are drugs or types of compounds used to stop infection by bacteria. Enzymes are biological catalysts. They speed up the rate of a chemical reaction. So what is PRDM2 and PRDM9? They belong to the PRDM family, and the PRDM family consists of the PR domain zinc finger proteins. The zinc finger proteins help to stabilize the folding of the protein. So basically, if you look at the picture up there on the upper right-hand corner, the green-yellow ball, the green ball in the center represents the zinc, and these items around it represent are the proteins that help hold the shape of the family. The full length of PRDM2 and PRDM9 are shown below. As you can see, for PRDM2, the characteristics of it include the PRDM domain and various zinc fingers. And PRDM9 consists of the triple-associated box domain, the PR domain, and a long string of zinc fingers. For my research project, I will be studying the only part of the construct of the full-length PRDM2 and PRDM9. For PRDM2, from 2 to 148 amino acids, and for PRDM9, 209 to 445 amino acids. They are represented by the red sections illustrated. So my objective and goal. So 1, to express and purify PRDM2 and PRDM9. Number 2, to test the activity of the enzymes associated with such proteins. I've only been able to work on the first one, and I'll present that today. So before we go any further, one of the most important protocols that I learned during our research was the preparation of buffers. Buffers were used in many of my experiments. So the first step in preparing a buffer is to weigh the solute on a digital scale. Second step, you need to measure the appropriate amount of deionized water and pour the solute into it. So basically, you add the solute to DD water. Third step, you place the beaker onto a magnetic stir, and you place the stir bar inside. So basically, that helps it to mix together. You wait until the solute is fully dissolved. You can see it is clear. And some buffers call for a specific pH. You can adjust that and measure that with a pH meter, and you can add KOH to make it more basic, or HCl to make it more acidic. And lastly, you must always remember to label the bottle with the name, date, pH, and concentration of your buffer. So the first step of my experiment was to transform the plasmids. So I took out the PRDM9 and PRDM2 plasmids from -80 degrees, and also the cells, and placed it on ice. I added 10 nanograms of plasmid and 10 microliters of cells, and incubated it on ice for 30 minutes. I heat shocked the tubes at 42 degrees Celsius in a water bath for 30 seconds each. Then I added the 200 microliters of stock media and incubated it to 37 degrees Celsius for 1 hour at 225 rotations per minute, revolutions per minute, excuse me. And basically the soft media helps facilitate the equal distribution of colonies in a plate. So the next step is to plate 200 microliters of the plasmids and cells, and basically I put it on an antibiotics plate and spread it around with a turntable. Lastly, I incubated it overnight at 37 degrees. Okay, next step, inoculation. I took 8 milliliters of LB meter media, and I added clindamycin sulfate and chloramphenicol. Both of them are antibiotics which are used for resistance to the, well, clindamycin sulfate is for resistance to plasmid, and chloramphenicol is for the resistance to cells. I added both antibiotics to the media, then I took 1 colony of the cells that I grew overnight and I placed it in the media as well. After that, I incubated overnight at 37 degrees, 225 revolutions per minute. Expression. From what I incubated, I had 8 milliliters of that sample. I took 2 milliliters and placed it in a glycerol stock solution. Then I put it on dry ice and then -80 degrees. Basically, for that, I did that in order to save time later on if I make another, if I express PRDM again, I can just get it from the stock solution. Then I took the remaining 6 milliliters and put it in 1 liter of LB Miller media containing the antibiotics. Half is sterile, so I put it in autoclave, which is intense heat and pressure. I incubated at 37 degrees at 225 RPM. Okay, so basically, this is okay. So after I incubated it, I took a sample of whatever incubated and found out the density using the UV visible spectrophotometer. And that actually measures the density of the sample. So after 1 hour and 20 minutes, I found the OD600. OD stands for optical density, and 600 is because I used, there was 600 nanometers of wavelength that passed through the sample. For PRDM2, it was 0.03, and about the same for PRDM9. And there's an X, as you can see, because I want the sample between 0.4 and 0.6. That's the ideal density of the sample that you want. So I did it again for 55 minutes. I didn't get the results that I wanted. I did it again for 53 minutes. For this, it's basically trial and error because you can't really predict how much it'll go up. Since it's not linear graph, you can't do it. It grows exponentially. So after 53 minutes, I found the correct, I found my results that I wanted. Then I cooled down the sample at 4 degrees for 15 minutes and add 4 micromolars of IPTG. The IPTG basically induces the expression of what I want. Then I incubated at 17 degrees Celsius overnight at 225 RPM. The next day, I centrifuged that, the 2 flasks, one with PRDM2 and one with PRDM9. So the last step is the purification of proteins. First step, I prepared the lysis, elution, and washing buffers. I added 10 milliliters of the lysis buffer to the cells that I had centrifuged overnight the night before. Then I French pressed the cells 4 times each to break them apart. So basically right now, you have the cells and a lysis buffer, which help break down the cell walls a little bit. If you put it in the French press, you can actually tear up the cell and all the parts will come out. So when all the parts come out, the only thing you want is protein. That would be held, the protein would be held in the supernate, which is the liquid. It will be free floating in the liquid. And at the bottom of the sample that you just French pressed will be all the rest of the cell parts that you don't know. You transfer supernatant, which is the liquid with the free-floating proteins, and you transfer it into a nickel column and shake it for 1 hour at 4 degrees Celsius. The nickel column helps to, well, that's the nickel column, and you basically, first you discard the contents of the liquid, you add the supernatant into the nickel column. And basically the nickel column has nickel beads, and these beads help these beads allow the proteins to be binded to it. So you pour everything in, all the proteins will be stuck onto the nickel beads, and the liquid flowing through, which we can discard because those are the rest of the media and other stuff that we added. You add 10. I wrote bead volumes over there to make it simpler, but they're actually called bed volumes. Basically, it depends. You add the amount of buffer that it corresponds to your amount of beads that you included. For me, it was 10 milliliters of bead volume of washing buffer. And basically, the washing buffer allows all the items that are not proteins to get flushed through one more time. And I collected 3 samples of the liquid flowing through, of the washing buffer. I labeled it W1, W2, and W3. Then I eluted the protein with 5 bed volumes of elution buffer. And basically, the elution buffer allows the proteins that were binded to the nickel beads to come out. And I collected 5 samples of each for each PRDM2 and PRDM9. And lastly, I ran the SCS pH gel, which is basically, I ran the gel to see how well the purification went. Okay, so the results. So if we look at the left, the first insertion right here, the first section, shows the first thing I inserted was a marker, which is like the control group. And basically, it shows the levels of molecular weights. KD stands for kilodaltons, and that's a unit. For PRDM2, as you can see, we had W1, W2, W3. Those are the washing buffers. As you can see, we don't have much protein, which is good, which means that we added enough beads for the proteins to be binded to the binding to the beads, so that none of the washing samples would have proteins in it. Then, if you look at the E1, E2, E3, E4, E5, which are the elution buffers, those are the, samples. Those are the ones that have the proteins. If you can look at the amount of, if you see the actual picture right here, I'm pointing at, that's not a mistake. That doesn't mean that when I ran the gel, something happened. There was so much protein that it was, like you can say, super saturated. It was so much that it was so purified that now there's a big glob, as you can see. And it's about correct, because the PRDM2, which is the left side, the molecular weight is 18.9, and it's right below number 20. If you look at PRDM9, the molecular weight is 29.1, and that's right where the arrow is. This one is about 50% pure, as you can see, because some of the proteins ran a little above.